Okay, so this is the final MCBHD lecture, chromosome abnormalities and the clinical consequence. Let's begin with the chromosome structure. So, DNA in the cell is not free, it's present as chromatin. Chromatin are the beads on the string, and the nucleosome are the building blocks of chromatin, or the individual beads. So, chromatin are beads on the string, nucleosomes are the individual beads. Chromatin can condense to form a chromosome. Chromatin can condense to form a chromosome. And here are two examples of chromosomes. So the above in the pink, we can see an entire mitotic chromosome made of two sister chromatids. So this is an entire chromosome made of two sister chromatids, attached at a centromere. They are identical, so the sister chromatids are identical, as the cell is going to divide and one chromatid is going to go to each of the new cells, along with the homologous pair in mitosis and not meiosis. So to describe this in other words, this two sister chromatids, this sister chromatid is going to separate from this sister chromatid is going to go into one cell, and the other sister chromatid from the other homologous pair is going to go into the same cell, and then these two sister chromatids are going to go into the same cell, and that is in mitosis, not meiosis. We have 23 pairs of chromosomes. So we have 23 pairs of chromosomes. So these, basically, we have 23 pairs of them. Yeah? And during mitosis, each pair will duplicate to form two sister chromatids, and that's how basically new cells and new genetic information is made. So genetic information can be split between the two cells. The additional pair, the first 22 are called autosomes. So the additional pair are X and Y, which are sex chromosomes. XX is female, XY is male. Okay. And this is a picture of a karyotype. So this is all our chromosomes laid out together. So these are what chromosomes in any cell will look like. If you were to take one of my skin cells and to show the karyotypes, it would look like this. However, when we're going under mitosis, here we have, this is a homologous pair. This will duplicate and this will duplicate. So this is from our mother, this is from our father. The one from the mother will duplicate and then will form like this kind of X, as we saw in this picture here. And the one from our father will also duplicate. And then those duplicated ones, there'll be like two X's. So this will be one X, this will be another X, and then you'll split them. Yeah, they'll be split and they'll be lining up. So this will line up at the equator, they'll both line up at the equator, and then they'll both split. Cool, carrying on. The basics of mitosis and meiosis. There are two different ways a cell can divide. Number one is mitosis. It's how the majority of cells in our body divide. It contributes to our growth and healing. Mitosis produces two daughter cells that are genetically identical to the parent cell. Two daughter cells that are genetically identical to the parent cell. The parent cell has 23 pairs of chromosomes, and so does the daughter cell. Which are an exact replica of the parent cell. So the parent cell has 23 pairs of chromosomes, and so does the daughter. And the daughter has an exact replica of the parent cell's chromosomes. The daughter cells are also identical to one another. So parent cells produce daughter cells. Daughter cells have an exact replica of their parents. Daughters are an exact replica of their parents, and the daughter cells are identical to each other. Simple. Number two, meiosis. So it's a type of cell division used to produce gametes and involves a reduction of genetic material in the cell. The parent cell has 23 pairs of chromosomes. However, the resulting four daughter chromosomes have 23 chromosomes only. These 23 chromosomes are also different to the parent cell, and each daughter cell is different from the other daughter cell. So the 23 chromosomes are also different to the parent cell. So the 23 chromosomes in the four daughter cells are different to the parent cell, and each daughter cell is different to other daughter cells. Easy peasy. This is because of crossing over, which occurs twice occurs twice during meiosis in metaphase 1 and metaphase 2. So look at the picture in notes. Let me see if there's a picture for you. Here we are. Metaphase 1 and metaphase 2. Simple. Moving on. Why do we look at chromosomes? They're responsible for developmental abnormalities and childhood growth. They're responsible for history of recurrent miscarriage and neonatal death. They can be responsible for infertility. Pregnancies can be predicted to be increased risk for miscarriage or disease following a genetic screening. And family history, it's good to look at family history, e.g. for things like cancer. So how do we look for chromosomes? This has been covered in other lectures as well, but we'll look into it again. So how do we look at chromosomes? There are many ways of looking at chromosomes. One way is karyotyping. The process of karyotyping is we take a blood sample, we culture the cells or the lymphocytes. Lymphocytes are a type of white blood cell that is used that in high amount it's a type of white blood cell that is high amount in the lymphatics. So lymphocytes are a type of white blood cells that have a high amount in the lymphatics. We arrest the cell in metaphase by adding colchicine. It needs to be in metaphase as DNA is most condensed in metaphase, so the chromosomes are easy to see. We then add hypertonic solution to lyse or burst the cells and release the chromosomes. So we're trying to release the chromosomes without them being damaged. We then mount the chromosomes onto a slide. There are different chromosomes identification methods we can then use. We can use G banding, which the G stands for chiasma stain. The chromosomes pick up a stain according to the amount of H-chromatin and U-chromatin in the DNA. This allows us to identify and compare people's chromosomes using banding patterns, which is called a basic karyotype. So G banding is a basic karyotype, and we can identify and compare people's chromosomes using banding patterns. The other techniques are FISH and array CGH, or array comparative genome hybridization. So to start off with FISH, we have already learned these, but we'll go into further detail, or we will supplement our learning. So FISH stands for fluorescence in situ hybridization. It's a method where we look for a specific sequence of DNA on one specific chromosome. We look for a specific sequence of DNA on one specific chromosome. We use a specific DNA probe that we make to identify the region of interest. We can see whether this region has been deleted or if it has been duplicated. And we'll show this in picture if I can get it up. So, for example, there's a child suspected of having a little chromosome 6 missing. We would do a karyotype, but the karyotype may not pick it up, as the area missing may be very small. So instead we do a FISH. However, with FISH, we need to know what we are looking for, so the lab can make a probe for it. We need to know the exact location of what we're looking for in the chromosome, so the lab can make a probe for it. The probe will be labeled with a fluorescent tag. We can then treat the patient's chromosome with the probe, and it will tell us if the region is there, or if it's missing, or if it's duplicated. If things are normal, there'll be 2 fluorescent tags. If there's a deletion, there'll be 1 or none tags, and if there's a duplication, there'll be 3 or 4 tags. And I'm going to show you this in the picture. Hope you can see it. Oh, the zoom is very bad. Oh, it's really bad. I'm really sorry, but there you can see. As you can see on the chromosome on the right, you can see there's 4 probes. As you can see on the chromosome on the left, there's only 2 probes, therefore indicating that part of that chromosome is missing. Easy peasy. We'll then move on to array CGH. So, if the patient came in and we had no idea what the genetic issue was, this means we can't get a special probe to be made. So we cannot do FISH. This is where we use array CGH. Instead of looking at the portion of one chromosome, this technique uses probes which are complementary to the entire genome. We use 60,000 probes, wow, 60,000 probes, all of which have a fluorescent tag and will bind to a bit of the genome. 60,000 probes, damn. This is what is called a hypothesis-free test. So technical, as we can do the test without having an idea of where the issue is. It's a hypothesis, free test. Chilling. Let's move on to chromosome abnormalities. So, beautiful picture. As we can see, zoom is shocking. We can have syndromes due to aneuploidy, chromosomal rearrangements, or microdeletions. So starting off with aneuploidy. So aneuploidy is an abnormal number of chromosomes in a cell. A diploid cell contains two sets of chromosomes, or 2n, which are 46 chromosomes in total, in a normal human. What you find in normal somatic cells. So 2n diploid cell, 2n, two sets of chromosomes, 46 chromosomes in total, and it's what you find in a normal somatic cell. So any skin cell, you'll find 46 chromosomes or 23 pairs of chromosomes. Easy peasy. A haploid cell contains one set of chromosomes, or n, just one n. So this is also 23 chromosomes in total, and what you'll find in a normal gamete, or any sex cell. In mitosis, the parent and the daughter cell are diploid. In meiosis, the parent is diploid while the four daughter cells are haploid. That's so important. In mitosis, the parent, and the daughter cell are diploid. In meiosis, the parent cell is diploid, where the four daughter cells are haploid. Easy peasy. So, aneuploidy are syndromes where a person doesn't have 23 pairs of chromosomes, but instead has an abnormal amount of chromosomes. So the person has an abnormal amount of chromosomes. As we said at the top, aneuploidy is an abnormal amount of chromosomes. Polyploidy is when the number of chromosomes you have is a multiple of 23, or a multiple of the haploid number. A multiple of the haploid number, or a multiple of 23. The haploid number, or n, is 23 anyway. So polyploidy is when there is a multiple of 23. So the number of chromosomes you have is a multiple of 23. So let's compare quickly aneuploidy versus polyploidy. So here is our normal gene, with a normal cell, where there's diploid 2n. In polyploidy we have, for example, we may have triploid, which is 3n. So therefore you have 23 times 3. So that means you'd have not 23, not 46, but you would have 68. You'd have 69. 69? 23 times 3 is 2, 4, 6, 60, 3, 6, 9. 69 chromosomes. Sorry, my maths is shocking. Or you could have tetraploid. So that wouldn't be 69, it would be 92 chromosomes, if I'm right, and forgive me if I'm wrong. So that's polyploidy. So that's where the number of chromosomes you have is a multiple of 23. However, aneuploid is just an abnormal number of chromosomes. So you might have trisomy, and in this example is trisomy 2, where there's 3 chromosomes of chromosome number 2. Or you may have monosomy. In this case, monosomy number 1, and this is when you have one chromosome for chromosome number 1. So in trisomy, it's chromosome 2 equals 2n plus 1. Monosomy, chromosome 1 equals 2n minus 1. Easy peasy. So how does aneuploidy arise? There's two ways in which aneuploidy arises, and that's the two ways we're going to investigate now. Number one, non-disjunction. Non-disjunction is the failure of homologous chromosomes or sister chromatids to separate normally during nuclear division, and if this occurs, then the gamete may have the wrong number of chromosomes. So if this is occurring when we're creating gametes, then the gametes may have the wrong number of chromosomes, and that's a disaster. So as we can see, here's normal. So this is normal separation of the chromosomes. Easy peasy, and that produces 4 daughter cells with haploid number, each genetically different. Easy. However, here we see a failure of homologous chromosomes, to separate in meiosis 1. Here we see a failure of sister chromatids, to separate in meiosis 2. Cool? And that means when that one gets fertilized, there'll be 3 of that chromosome, and when these two get fertilized, there'll be 3 of each chromosome, and when these get fertilized, there'll only be 1 of each chromosome. So that would be trisomy, that would be monosomy, that would be trisomy, that would be monosomy, that would be normal. Easy peasy. So an example of this is trisomy 21 or Down syndrome. This is the most common aneuploidy. It's a non-disjunction. It's non-disjunction